So there are two different arguments I'm going to touch today. One is a discussion on the rate of adaptation, or if you like, the speed in which evolutionary processes take place. And the other one is a way of connecting the population genetics to what is actually measured in experiments in which you go around in a population and you pick up some samples, and then you try to see how these samples differ from one another, what is the distribution of genotype frequencies. Usually you don't take a genotype frequency of the whole genome, because in that case you would have a great deal of singletons. You're looking at some loci. In these loci you find a number of segregating alleles. You ask you can measure what is the frequency of your segregating alleles in your sample, and you try to infer from that something about what this population is being going on. We'll do that in a simple case, which is a neutral case, and I will show you what, say, just the direction, what happens if instead you have a selection. Of course, what you actually do in actual work is doing the reverse. You take your sample, you find some frequency distribution, and you're trying to imagine from this frequency distribution, what are the selective forces that act on it. And I will not be able to discuss pretty much this. I just will try to open your curiosity on this problem that will probably be dealt with in the next 2 weeks. So first of all, speed of adaptation. Now, let us consider first, in this talk I will consider only haploid asexuals population. Imagine that you have a population in which there is, say, a population of fixed size with some mutation rate. This is mutation rate for the locus we are interested in, but you can imagine that this is, say, you are in the limit in which the population size times the mutation rate is much less than 1. So what you expect to happen? Well, you wait, and then after a while, you find that there is a new variant arriving by mutation, by the mechanism that I discussed yesterday. And if this mutation is advantageous, then there is some probability that we have, we didn't compute, but I showed you yesterday, which is non-zero, that it goes eventually to fixation. So schematically, you can imagine that this is just the cartoon of the amount of individuals in the population which carry this variant. So, and they, of course, carry this variant because they share the ancestor. The ancestor is the one of the guys in which mutation, this variant arose by mutation, and then they reach fixation, so we have a big A and so on. And then after a while, a new variant B arises, and if it is advantageous, you might have that, it eventually fixates and so on. And so in this way, you can go on, one advantageous mutation fixating after the other. And you can imagine that in this regime, in fact, the speed of adaptation will increase roughly proportional to the population size. Basically, because if you have a larger population size, the number of mutants which arise at each generation will be proportional to, it's n times mu, so it will be proportional to population size. So in fact, what you have is that if your population doubles, the time interval between these new mutations which arise, and each of them has the same probability of going to fixation, will decrease by a factor 1/2. So if you double the population, will decrease. Yes, please. Mu is the mutation rate, that is the probability that an advantageous mutation arises per individual and per generation. N is the population size. So n times mu is a parameter which essentially tells you the expected number of advantageous mutations which arise at each generation. Okay? So if you have no more than one mutation per generation, then you can expect that these mutations arise one by one, and then they have a certain time. We evaluated. You remember, we evaluated the time to fixation. The time to fixation is of the order of log n divided by the fitness advantage, divided by S. We'll use that again. So if you imagine that this is such that the time between the appearance of an advantageous mutation is longer than the time, the expected time for fixation of this advantageous mutation, we are in this regime which is called the periodic selection, in which the rate of adaptation, that is the rate by which new advantageous variants are fixated in the population, is proportional to the population size. Is that right? So this is a simple regime, and so you see that in this simple regime we expect that the rate of adaptation is proportional to the mutation rate and to the population size. Now, what I'm going to argue is that if your population increases beyond a certain threshold, if you like, then the rate of adaptation does no more increase proportionally to n, but increase more slowly. And let's see why. Well, what happens is roughly, so this is a concept that was introduced by Gerish and Lenski about 20 years ago, called Clonal Interference. So the idea is that, well, you start from your wild-type population, and then you have this nice-looking, advantageous variant which arises in the population. And then, lo and behold, you have that there is another advantageous mutation which arises in the population, starting from the wild-type. But meanwhile, this A variant is going to fixation. So that means that this new mutation has arisen in the wild type. But now the people who who dominate the population are the children, are the descendants of this guy here who was lucky enough to get the A mutation before. So for all practical purposes, this nice mutation C or B, which would be advantageous, are lost. Whereas if you had waited, no? You see, you had waited for it to appear in a descendant of A, then B would go to fixation. But maybe here you have that again, C appears in one of the guys who carry the A variant, and this nice mutation is again lost until you have to wait for it to appear in the descendants of these AB guys, and so on and so forth. So you have that some advantageous mutation arise in the population by mutations, but they are lost because they are competing with segregating variants which are fit enough and they are going to fixation. Right. In fact, what can happen is that even that this C may be by itself, could even be, say, more fit than A, but it doesn't succeed in getting to a fraction of a population large enough to reach fixation. And if it does it, I mean, if it does, imagine that you have a situation in which a C is better than A. And then that by some chances, C succeeds in growing beyond, you remember we had this threshold, N star, beyond which we would say that the variant was established. If C goes beyond N star, then it simply dislocates A, and you would have only C and not A. So there will be a fight between the different variants, and this is due to the fact that we have clones. We have asexual reproduction, so the genome reproduces as a whole. Imagine that you have instead a population which reproduces by sexual mechanisms so that you can have a recombination. Then in this case, you might have that the locus in which C appears is different than the locus in which A appears. And then by recombination, they could find themselves together in the same genotype. This genotype will now have the double advantage, and then it will be much easier for it to spread the different alleles in the population. And this is one of the arguments that people use to say that there is an advantage for sexual reproduction in spite of the obvious fact that in sexual reproduction, in fact, you can say that there is half of the population that in some sense does not contribute much to the overall fitness. No, you need two individuals where before you needed only one. So there is a price for having two sexes, and this price must be balanced by a very strong evolutionary advantage. Unfortunately, I'm not going to discuss about the why sex. It's a fascinating problem, but that would drive us too far. Okay, so let us remind what is the fixation probability of a mutant. Let's assume that we are in a situation in which the mutant appears in a population of size n with some frequency x0. And then we know we have, we haven't derived it, but I just told you that the probability of going eventually to fixation is given by this fundamental formula. So 1 minus e to the minus 2n times s, which is the fitness advantage, times x0, divided 1 minus e to the minus 2n s. And what I wanted to stress is that here, if you multiply n times x0, what you find is small n, which is the number of mutants in the population. So whenever ns is larger than or the order of 1, the probability of fixation is of order 1. And we say that the mutation is established. And if we have a mutant which arises in one individual at a certain time, then of course we have to put n equals to 1. Therefore, the probability of fixation of a mutant with the selective advantage s will be given by this formula evaluated for x0 equal 1 over n, which is given by this nice looking formula, which has, which we discussed a little bit yesterday. So you have essentially three regimes. One regime is the one in which s is, n times s is smaller than 1. Then this essentially tells you that the probability of fixation is 1 over n, as if the mutation were neutral. And this works both ways, for both advantageous and disadvantageous variants. If s is large and positive, so that n times s is larger than 1, then essentially what you have is that the probability of fixation is of the order of s itself. We are assuming that s is small, n times n is large, s is small, then as you expand this, you find that essentially you get only s. And then if s is large in absolute value, such that n times absolute values of s is larger than 1 and s is negative, then the probability of fixation is essentially 0. It's exponentially small in the population size. Okay. Now we wish to see what happens if we have intermediate size populations in which this effect of clonal interference, that is the appearance, the simultaneous appearance of different advantageous variants, takes place. So let's assume that, this is a simplification, that mutations arise independently from the wild type. So this is like the case I showed you before, in which we had A, B, and C appearing from the wild type. we are considering only advantageous mutants, essentially because we simplify the matter. And we assume that also that the selective advantage has some distribution. Now, this is again a sort of sea snake. There is an argument by Alan Orr, that this is the guy that Kavita mentioned this morning, saying essentially the following thing. We expect that organisms are already well adapted. So after all, they have survived, and they are survive and thrive in the environment. Therefore, for a variant to be advantageous, it must be some sort of extreme. It cannot be just a generic variant. It must be really something that is close to the tail of the distribution of the effects of variant. Otherwise, it will not be advantageous with respect to what is already established in the population. So the correct distribution that you would expect to apply for advantageous mutations is a sort of extreme value distribution. I don't know if you have seen any extreme value distributions. Have you ever seen this problem? So, Gumbel? Yeah? So you know what is the problem. The problem is that you have a sample, for instance, of a sample taken from a normal distribution, and you ask, what is the distribution of the best one in the bunch? And this distribution has a very nice shape because it is, on the one hand, on the lower hand, it decreases very, very fast. It decreases like the exponential of an exponential. And on the upper level, decreases like a normal exponential. All right? So we are interested in the upper side because we are interested in the distribution of advantageous variant. So we expect it to be like the Gumbel distribution on the upper side, and it goes like an exponential. And it turns out that mathematically exponential distributions are very nice to treat. So this is an argument that Alan Orr introduced to say that this rho of S can be probably will be something of the order of an exponential. I would say that experimentally, it has been evaluated in lab strain E. coli in several experiments, in particular by Isabel Gordo in Portugal, and it works. I wouldn't say very well, but nicely. You can accept as a rule of thumb that this distribution is some sort of exponential, where the rate of the exponential depends on what you're looking at and also on the environment. It's not the same distribution. If you look at the ordinary environment for E. coli, would be ordinary lab medium, or you in a poor medium, and so on. Okay, now let's ask, what is the probability of mutation of a probability of fixation of a mutant which has an advantage S with respect to the wild type? The fact of having as a benchmark the wild type is very useful. So what is this probability? Well, there is the probability of the mutation to arise, which is this rho of S. There is the probability that this mutation goes to fixation if nothing else happens, which is the pi of S that I showed you before. So this is plain. It would be the same if we had the periodic selection process. And then there is this factor that you see is a reducing factor, is exponential, and we see of a positive quantity. Over negative quantities, minus lambda S. What is this lambda S? It looks like yes, please. You can take it like that, but in fact, I'm not going really to distinguish between the two. In fact, also that in practice, it's very hard to say what is n and what is mu. Yeah. I'm considering, in this case, I'm considering mu fixed, and I imagine that the population is larger. Yes, that's true. But in practice, you always see the combined effect, as Kavita mentioned also. It's this famous theta, which is proportional to the product of n times mu. And we'll see it again. Okay, let's see what is this quantity here. This quantity here is related. So this factor tells you what is the probability that no better mutant arises and establishes. You remember, there is arising and establishing in the time that our guy goes to fixation. So it is proportional to the time needed to go to fixation, which is log n divided by s, right? This is one thing. Then you have this factor, n mu. N mu, what is it? Probability of having a mutation. And then there is this integral. This integral tells you, well, we are looking only at better variance, so we have to integrate from s to infinity. This rho is the probability that the mutant which arises, it arises with the rate n mu, is better than s, this rho x. And this pi is the probability that this guy goes to fixation. And you see, when I do that, I don't take into account the fact there might be other variants going around. So in this sense, it is a sort of a first-order approximation. We are just looking at the fight between variants two at a time. All right? And then, of course, so this is the probability of going to fixation. We want to have the probability of not going to fixation, so we have a 1 minus that, and we have to exponentiate because it can arise over time. So this is why we got this time. And now you see that here we get something that goes like n to the power minus n, something like that. So you see there is log n is under exponential times something that is of order n. So this reduces. And in fact, you see, if you look at that, it starts from n, and then it starts increasing more or less like log n after a while. So we have a very important reduction, not a reduction of the speed. The speed does not go down, keeps increasing, but increases much, much more slowly. So this is a sort of paradoxical. You would imagine that there would be a huge advantage having a very large population because you would able to fish for better variance. And in fact, you're going down. You are not increasing that much. Now, you can try to make this a little bit more quantitative in this way. So you ask, what is the expected fixation rate per generation? Well, so the number of fixation events that you have per generations will be given by the integral over all values of the fitness of the probability that a mutant, which in fact there should be also the probability of arising here, okay. So no, I put it here. Yeah, there is everything in there. So it's this probability of going to fixation. So it's given by this the number of this probability. So the total value of this fixation probability, and then if you multiply by s, you integrate, you get the total expected fitness increment, and if you divide by the number of substitution, you have the fitness increment per substitution. And with that, you can evaluate something that is the expected behavior of the Wrightian fitness. You remember Wrightian versus Fisher of Malthusian fitness. So Wrightian fitness is the expected number of offspring. So as a function of number of generation divided by the number of generations, so this is a measure of the rate of adaptation. It's called the rate of adaptation, RA. If you see this RA in the papers, this is what we are computing here. And will be given by this expression here, which is very easy to understand. You see, so this is, how to say, you have, this is the expected number of substitution per generation, and this is essentially what you have if you assume that there is a certain increment per substitution. You essentially, you want to have to, I forgot the log here, sorry, I forgot the log here. The change in the Wrightian fitness is given by 1 plus ES, and therefore the expected change in the log of the Wrightian fitness is log of 1 plus ES. That's all. And because I forgot, sorry, I forgot this log here. K is the number, expected number of substitutions. No, K is the number of substitutions per generation, and EOK is the expected number, the average number of substitution per generation, which is usually a small number. Okay? Yes, please. Yeah, usually it's a small number. Yes, that's absolutely, 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 yeah. Yeah, but then we see that we have to, this is absolutely true, but if you have large population, what you can have is that you have one substitution and many variants get fixed at the same time. We'll see just in a moment. I'm not making a quantitative theory. So this is what I'm going to discuss. This theory is very nice, but it works only at intermediate populations. So we say, small populations, we have periodic selection. Intermediate population, we have this clonal interference in which, you remember, the variants arise from the wild type and they fight one against the other. What happens if the population is so large that you have many variants? Now, many variants means that some of them arise from the wild type, and some of them, like we have seen, AB, arise from somebody who has already mutated and segregating. It's not yet in fixation. So what you end up is that you might have that the fight is no more against single mutations on the background of the wild type, but multiple mutations, one against the other. And this is a very fascinating theory that, say, for instance, there's this nice paper by Park and Krug, which I'm not going to discuss, but just imagine that what is going to happen is very nice. So if you have this kind of situation, then the fixation itself can be described in a different way. Because at any time you have anyway a great number of variants which are segregating. So what does it mean to have fixation? Fixation means that the whole population, this is a clonal population, changes its last common ancestor. And we will see it better than when we discuss the genealogies and the coalescent in the second part of this lecture. But in the fixation, so the last common ancestor changes suddenly, it can change quite a lot. You might have that several variants go to fixation at the same time. So we have to distinguish what is fixation at the level of genealogy from what is fixation at the level of variants. So, and also that the origination and fixation rate have to be distinguished. So because several mutations can fix simultaneously. Yes, please. No, it's not a hierarchy. In time, you have a last common ancestor, and then, okay, the population goes on for some times, and it remains there. Then, at a certain point, you find, lo and behold, that the last common ancestor is much more recent, suddenly, right? And then you wait a long enough, and then you jump to another one. This is what happens. So in a clonal population, you have that the last common ancestor is, and then one of the problems that we'll discuss in the second part will be what is, for instance, the expected age of the last common ancestor. Can you say something about that? What is the rate? So, which is related, of course, to the rate by which the last common ancestor changes, which is this fixation rate in this language. So what happens is that now we have seen that the interference between single mutations led to a decrease of the adaptation rate. But now you gain something because you can have a great deal of mutations which fix at the same time. So this leads to a relative increase in the adaptation speed. And also you can also find that the pace of adaptation becomes less irregular and becomes a little bit more regular. And this, I just wanted to show this paper from this paper of Park and Krug. The references are in the reading material after the. So what you have is, okay, so here you see this line here is the number of mutations in the most populated genotype. So that means this is not, because we cannot really say what is the, say the wild type is the most populated genotype in this case, okay? We cannot really say what is the wild type. It's not like in the other situation in which we had that the population was monomorphic, and then from time to time you would have a segregating allele, and then again becoming monomorphic. So, and this, as you see, this is pretty regular because the mutations arise with this rhythm that is essentially n mu, and the most populated genotype is essentially a finite fraction of the population. So this is a very regular pace, and it is this effect which makes that the adaptation process is more regular. You always know that something is going to happen in the most populated genotype. This is the number of fixed mutations, and what you see is that it's more irregular. You have these jumps. No, you have these jumps, and these jumps correspond to the fixation of several alleles, several variants in one go. And this is the number of fixation events, and this is, well, there is some irregularity here and there, you see? Some irregularity, and it goes, this is actually, if you, there is a mention in the paper that these are really two steps, but they are very close one to another. It's not that the two fixation take place at the same time, because that is not possible. But there is really just a very small delay. And one of the contributions of this paper is that they were able to simulate very large population with a, with a clever algorithm. Being the change in the last common ancestor. Fixation event is the change in the last common ancestor. Mm-hmm. It means that from the start of the simulation, the common ancestor of the population changes 5 times. Okay, so this is as a function of the population size, you see. And this is a relative error with respect to that formula that I gave you before of the adaptation rate in the Gary-Shelensky theory for. So this is for different quantities. EK, which is the number of expected number of fixation events. So this is the expected value of the fitness increment, and this is the rate of adaptation that I told you before, which is essentially given by that formula that I gave you, EK times log of 1 plus ES. And what you see is that the all these are larger than expected from the garish landscape would be 0, and the rate of adaptation shows, so this is the rate of adaptation. So it shows it increases by some factor, 20-25%, with respect to what you get from the garish formula. Okay, so this is, but there is some theory on that, but basically this is what you get from the from simulations, and then later how we can, what are the implications of this multiple fixation in the genealogies. And there again, I will only give you something to stimulate your appetite, because the theory is very nice, but is sort of beyond what I'm able to tell you at the moment. So let's go to the coalescent. What is the coalescent? No, it doesn't. This is the error. Yeah, I don't know. I mean, this is still positive anyway. Yeah, probably. I don't remember. I think that, anyway, this can be a fluke. It can be an error. Coalescent. Now, I like a coalescent because this is what got me started in the business. But so coalescent is a concept that is trying to answer these questions. How far in the past must we go to reach the last common ancestor of any individuals? You have a population, you pick up a sample, and then you ask, well, how far should I go to find the last common ancestor? I'm staying in the asexual case, and actually I'm really telling you only about the neutral case without selection, and I will introduce mutations later. So for the time being, we are only trying to make a mathematical theory of genealogies as a random process. How many different genotypes can we expect? This is very important. This is very important. So the idea is that if you have an asexual population, you pick up a certain sample, and then you ask, well, how many different variants should I expect? Can I infer something from what I get about the mutation rate, for instance? How do the times of last common ancestors depend on a particular chosen sample or on the population size? How do these quantities fluctuate as the population evolves in time? How are these affected by selection? So these questions are addressed by using the concept of the coalescent, which is due to this guy, Kingham. And the coalescent is a very simple and very surprising concept. We have seen that the Wright-Fisher model is defined in this way. Now, you remember, the Wright-Fisher model is a very simple model of discrete generations in which you have a population of size n with several variants, say white and black, and then you want to build up a new population out of that. And the way in which you do is, say, you let each of them reproduce as much as they can, and then you pick up at random n individuals. This is probably what happens in real world with a number of details that are very important and are completely neglected. But of course, this is not the best way to simulate it. It's much easier to reverse the process. That is, you have now n cases, and you want to fill them with descendants, with offspring of the previous population. So you pick up at random the parent, and then you copy it and you put it in the case. This is what Kavita mentioned. No? This is the way in which you actually go and simulate your process. And the idea of the coalescent is the following. Well, okay, then if I have a population here, it is the result of iterating this process back in the past, as long as to, say, since the beginning of the computer experiment, if you like. So what does it mean? It means that at each generation, what we are doing is an application of the label, the label of this of this new individual, for instance, this would be individual number 3, is mapped into individual number 1 of the earlier generation. And then we iterate this mapping, but picking up a new mapping at each generation as we go back. Now, what happens? What happens is that from time to time, 2 of these arrows will map on the same individual. So this, what we interpret this as being that this individual had the 2 offspring which have been reproduced long after that. But if you look at from the point of view of the individuals, say, of the population at the present generation, it means that if we go back and look at the lineage of the ancestors, then these ancestors collide or coalesce into a common ancestor at some time. And now the problem is that can we make some quantitative theory about that? So let's make it. So this is the idea. We have a population of size n, big N. Now, we don't want to describe the whole population. We want to describe what happens to a sample or small n, which is find a reasonable number that we can handle. So we have a certain number of individuals, 1, 2, 3, to small n here. And then we look back to their ancestors, and we look back at their genealogies, and we see that from time to time there are coalescence, and then there are some other coalescence, and so on and so forth. And then we make a genealogical tree. Usually we are accustomed to a genealogical tree which goes this way, don't we? But this is asexual, so the only thing you can have is reducing the number of ancestors. If we go back in our family, our ancestors grow, a number of ancestors grows exponentially until it reaches some constant value, which is our isolate is called. But for asexual, we know that it's going to reduce, and eventually it reaches the most recent common ancestor, MRCA. All right? Now, what we can say is, for instance, if we consider the individuals whose last common, with a subset of these individuals whose last common ancestor existed T generations ago, we can call it a T family. So one of the questions is, how are the sizes of T families distributed? All right? So this is what the Coalescent is going to answer. And you see that you will see that it is a very simple theory and very useful. You remember, this is a null model. Here, all the individuals are equivalent, so there is no selection, and for the time being, there is no mutation. So this is very, if you see deviations from what this theory is going to tell you, it means that something is going on. Yeah, only that the rate of coalescence depends on how many particles you have. So it is non-trivial. So we are considering a neutral Wright-Fisher process. That means that there is no selection. So set T equals 0 for the present, and then we go back in time. So T is the number of generations ago that we are considering. We can label the individuals from 1 to big N, say, and at each generation we define application P, which maps individual I to its parent in the immediately previous generation. So parent and ancestor is a parent or a parent of an ancestor, right? Now, this application is extracted at random independently for each I and each T. Just a mapping to, say, from I to one label between 1 and n. And the ancestor at T time is just the application first of the application 1, then of 2, and so on, until T generations ago. Is that clear? For the statistical physicists, this is called an annealed mapping process. Annealed because it is extracted at each time is different in general. Then we define a lineage. The lineage is just the sequence of labels starting from now to the ancestor, through the ancestor, going back in time. So this is just a lineage in genealogy. Remember, for instance, in the royal families in which the throne was only given to males, that you would have a lineage of males, you know, in principle. And then lineage coalescence happens when you have that the ancestor of individual I at time t is equal to the ancestor of the individual j at time t, assuming that before they were always different, and I is different from j. And the coalescence time is the, say, the smallest value of t for which this happens, okay? Is it clear what is a coalescence? So this is the smallest value because then, because for t minus 1 you have that the ancestors were different. So this is fine. In this way we have defined the coalescence time and the common last common ancestor of I and j. Well, this is probably not necessary. So now we can ask, how many generations are needed to reach the most recent common ancestor? What is the distribution of the coalescence time? What are the consequences for quantities that we can measure? Now, this is a point which is interesting. If instead of looking at individuals, we look at genes, by and large, you can imagine that for a single gene, for instance, for a single sequence in a gene, you can neglect up to a certain point recombination. The reason is that if a recombination happens, so to split a gene and to glue it in a different way, usually the gene becomes useless, and so the corresponding individual will get out of the population. So usually you can imagine that the variants of the gene up to a certain point are going as clonal. So for a sequence which is sufficiently short, you can still apply this mathematics as if it went on as a clonal process. So what you can speak is of gene genealogies. So that means that what we are saying now for haploid individuals and so on and so forth applies to some extent also to genes, with the only difference, which is important but trivial, that because we are speaking of diploid individuals, you have 2 copies of the gene, so the population is twice larger. Right? This is why it's not just an exercise, but is something that is useful also in actual genetics. Other disclaimer, effective population size. This was mentioned also by Kavita. And so whenever you see n is not the actual population size, it's the effective population size, there's a problem with the effective population size, which I'm going, hopefully, to discuss later. Yeah? Who's that?
N equal to 2 that you said when talking about diploid, coming from the haploid. Mm-hmm. When we are taking the diploid case.
I'm not discussing the diploid case because as far as I'm concerned, they behave like haploids with a twice larger population.
But my question is,
yeah.
For diploid, there are 2 alleles.
Yeah.
Suppose A and B, and there can be total 4 combinations. AA, AB, BA, BB.
No, but this is what I mentioned. I'm looking at one single locus, and then this single locus by itself undergoes some coalescent process, and as long as it is not disrupted by recombination inside, I can treat it as clonal. This is what I'm saying. So I'm not looking at the linkage between these different loci at this level. So this is why I can, as far as I can look at the, say, a short enough stretch of the genome, which is not necessarily a gene, then I can use these mathematics up to a certain level. So what I'm neglecting is the possibility something happens in the middle, which is probably say, a probability relatively small. Of course, there is research on what you can do beyond that, but I won't touch on it. If you like, there is a book by Wulff and other people called Genealogies, in which it discusses it and becomes rapidly. And let's distinguish two things. One is the label in the population, which is, let's put it this way. We start with an initial population, and there I have individuals which I will label with the letters of the alphabet. We have an alphabet like in China, no, with a great number of letters. Okay, so each of them has a different Chinese letter. And then they occupy a certain number of cases, and the cases are labeled from 1 to n, right? Now, next generation, we have n cases. And then for each of these cases, I pick one of the individuals from the earlier generation at random and copy its letter and put it here, right? And then I iterate the process. In this case, the Chinese letter will get lost. And eventually, fixation would be that all the individuals have the same Chinese letter. Is that clear? So this is the process. But the case numbers, the number of cases, has no meaning. Absolutely no meaning. So this is what I mentioned here. If we have that there is equal fitness for all types, so the process is neutral, there are no subdivisions in the population, geographical or otherwise, and we have constant population size. So with first two assumptions, tell you that there is exchangeability. Every individuals can be exchanged with any other individuals. There is nothing special about it, except that we can label it by a Chinese letter. And then we can see what is the frequency of Chinese letters around. So the number of offspring of any individual is statistically the same random variable as for any other individual. And you can exercise what is the distribution of offspring of an individual. I won't make this exercise for now. You will make it in your head. And now we look at the problem, say, the statistics of the coalescent. Looks very nice. So let's ask, what is the probability that n individuals have whole different parents in one generation? So what is this probability? Well, the first individual can choose whichever parent he wants, no problem. The second will have to choose a different one. So there is 1 minus 1 over n probability. The third will have to choose one which is different from the first two, so the 1 minus 2 over n. And eventually we have 1 minus n minus 1 over n for the last one, right? And then we assume that n is finite and big N is very large. So we can just simplify this. What you get is the sum of 1 over n divided by the sum of the first n minus 1 numbers, small n minus 1 numbers, and so it becomes 1 minus n times n minus 1 divided by 2n. Is that right? Now we ask this question. Imagine that we have n individuals, we go back t generations, and then we ask, are the ancestors of these individuals all different? Well, call this probability that this happens, pi n of t, and then we can ask, well, this is very simple, because we know that what happens if the n individuals t generations ago were all different, the ancestors t plus 1 generations ago will be different if they were different at this time, and then if their corresponding parents are all different. Is that right? So what you have is that pi n of t plus 1 is equal to pi n of t times this wn that I have evaluated here. So you can put this expression here, and now we can exponentiate, because this quantity is always a small quantity. So, and we have a nice exponential e to that the pi n of t, which of course is equal to 1 when t is equal to 0, because these are different individuals, is equal to e to the minus n times n minus 1 times t divided to n. And in particular, if we pick up two individuals at random and we ask, what is the probability that their ancestors t generations ago were different, is given by e to the minus t over n. So what we have learned out of that is that the scale in which the coalescence takes place is proportional to population size. Okay? And now this means that this is a slow process. Population sizes are large. So just keep that in mind. Now, we want to make statistics of that. And here we have to distinguish two things. So we are given a population. A population is a sample of the process at a given time. And there is something of the process. So the process has itself some distribution. So I want to distinguish what happens when we take the average of one population, one sample, which is actual sample, maybe of an actual population that I'm studied, and the mathematical average. Now, there is something nice to say that the mathematical average, if you imagine that you are able to follow this population for a great, great number of generations, which is many times its population size, then we can average over time, and this will be the same as averaging over the process. In this sense, this is ergodic for people who like ergodicity. You see the problem. If we are doing this with a bacterial population of the order of a few billions, we should have to wait a few billions generation time. And of course, well, it's true that generation time is 20 minutes, but billions of 20 minutes is probably longer than your PhD allowance. So I will distinguish the average of the process by the overbar and the averages of the population by the angular brackets. So if you take this formula that I just derived here, this one, what you get is that the average of the process of the age of the last common ancestor of two individuals picked at random is equal to the population size, which is, I think, a nice and clean result. A time to have the same ancestor, right? And then I average over the process, and this is equal to the population size. Now, what are now the implications? Let's just take an infinite site model for the time being, with a mutation rate U per genome and per generations. So we can ask, what is the expected number of mutations between me and my last common ancestor with VJ? All right? So we have, what am I doing? Up. Okay. So the expected number of mutations with respect to the common ancestor will be equal to n times u, where u is the rate of mutation per generation. And therefore, if I compare the number of mutations that I have with the number of mutations that VJ has, on average, will be twice this number. And we find again that the 2nu, you remember that Kavita derived. This is what was called theta. For a case of diploid, it would be 4nu. So just careful when you read the literature. Now, what I wanted to show is the following, that if you look at So here we are looking at a population of 50 individuals, and we just look at what is the distribution of the age of the last common ancestor between any two pairs. And this is the kind of things that you find, something like that. But this is a generation 200, this is a generation 300, this is generation 400, 500. What you see from there? Can you tell me something about this kind of distributions? Which are the obvious things you notice in these distributions? First of all, they all look different. No? They are widely changing in time. Second, something is common, however. What is common? More crowded near the origin and more sparse, and it will be higher peak. Okay, so can we? The height. Well, the expectation value we say should be around 50. No, the population is 50, so it's around here. No, okay. The expectation value itself fluctuates from time to time, yeah? You expect. But the expectation value of the expectation value, if you like. Well, I would change slightly the terminology. If I'm looking at something that is defined by the sample, I call it the mean. If I'm looking at something that is defined on the process, I call it average. So the mean fluctuates, and the average should be 50 for this one. All right? Question? Okay. This is something like an aside. I told you that there was an exercise, evaluate what is the probability distribution of the number of offspring. Now, in this case, it's very easy to answer because the distribution must be a Poisson distribution with average 1. Why? Population is fixed, so the average must be 1. Why Poisson? Yeah, because I say it's independent for each of them, and for each of them is a small number of order, no? Must be, yeah, because if we have n trials, and each of them has only one possibility, okay, 1 or 2, say, or something. So we define the average number of offspring as being, of course, a sum over number of offspring, probability of them, and this must be equal to 1 simply because the size of the population is invariant. Now, what is the probability of coalescence for n lineages? And this is given by 1 minus that Wn that we have evaluated before. And you just plug it in this case, we have this pi of m. This is the number of pairings that you can make with the lineages. This is the number of offspring, and this is the probability that these guys just jump on the same parent, isn't it? No? It is probability that this parent has m offspring. Looking backwards means that we have m ancestors that suddenly coalesce into 1. And so you can evaluate this. This is given by this n minus 1 divided to n, which is what we got before, times what? Times the average value of m squared, you see here, minus 1, which is the average value of m. All right? So this is probability of coalescence for n lineages depends on this average value of m squared. Now, define this quantity, m squared minus 1 is equal to kappa. So I just restate this result. And now we have two possibilities. One possibility is that this quantity is finite. If this quantity is finite, I can prove it, or let's put it this way, Derrida and company proved that all results hold that I have given you up to a rescaling in time, which is always of order n anyway. So kappa is finite as well. So you can use the same, always can use the time units so that Wn is of the form that I gave you. Right? So what is the other case? The other case is the one in which this guy is infinite. So you have a very, very wide spread of the number of hosts. And now I advance you 1 point. The result of the neutral genealogies hold for a great number of cases, as long as this coalescence is, so this variance in the number of offspring is finite. Whenever you introduce, say, selection, you will have a different statistics, and this different statistics is due to the fact that you have a multiple coalescence with slowly decreasing probability. And in this case, you go to a different behavior, which is also universal, that I will show you. I will not derive you. I will just show you. I will discuss a little bit about that. So what you expect is that there is a great difference between the neutral and the selection. In reality, what you have is an intermediate case, in the sense that selection is there, but is not so strong to, say, throw away whatever we are saying. And one of the reasons is that the selection takes also some time to act. Now let's go more in detail. What is the probability of a genealogy? We have got this Wn, which is important, with this pi of n. So let's imagine that we have a genealogy. Now the genealogy is not only the topology of the tree. We see that the topology is not so relevant. What is important for us is the time delay between the different coalescent times. So the distribution of the coalescent times. So here we start with seven individuals at generation 0, and then we have a first coalescent, and this is tau 7, and then 6, and then 5, and so on, until we go to the most recent common ancestor. So we're given this collection of times, and we ask, what is the probability that the genealogy has this distribution of coalescence times? Well, it's given by this pi. You simply have to multiply this pi. So you have a pi 2 of tau 2, then pi 3 of tau 3, and then a pi, and so on and so forth. And if you plug in the evaluation, so what we get is that each tau k, each coalescent time with k lineages is an independent random variable with the distribution given by this expression. Always measuring generations in unit of n, which is the population size. I rescaled that, okay? So this is one. Now, out of this, you can get lots of interesting. So distribution of the age of the most recent common ancestor for n samples. It will be given by the sum of these random variables. Each of these is exponentially distributed with this average, and they are independent. So it's a sum of independent. The average of the sum of the independent variable is very easy. And you can also look at the probability distribution, which gets, say, this is very simple. This is just making explicit what I was saying. There is a little bit more of algebra, but it's essentially the same thing. You have, the only thing is that we have to keep the sum fixed and then take into account all the possible ways in which you decompose it. I would like to say rather to make this calculation via the generating function. But anyway, Tavare and Ney did the calculation like that, and they have a nice explicit formula. So why not? Now let's plug in again mutations. So far we have just calculated the average of mutations between two individuals taken at random. Now here we can get something which is more interesting, which is the probability of mutation. So what is the distribution of mutations in a sample? And this is much more important. So coalescence mutations, so this calculation I have already done, I think. So, and we get that the probability that you have the same, so that you end up with the same, is given by this 1 divided by 1 plus theta. But let's go, what happens if we have a samples from a large population with a given value of theta, and say that samples belong to the same group if they happen to have the same allele. Now, I am going from the infinite site model to the infinite allele model. Whenever there is a mutation, it's a new allele, right? So this is slightly different. Now you ask, well, okay, if I don't look finally into the structure of the alleles, I say they are either equal or different, this is the infinite allele model, then I bin my population in groups which have the same alleles. And then I ask, what is the probability that I have B1 groups with singletons, B1 singletons, B2 groups with 2 individuals, B3 groups for 3 individuals, BK groups with K individuals? Is that okay? So I just ask you, what is called the frequency spectrum of the alleles? This is a very important thing because you can actually measure it. And you can infer from what you measure. First of all, if it corresponds to the neutral case, you can infer the value of theta from what you measure, right? And if you see that the frequency has a different distribution, then you can infer that there is something else going on, probably that there is some selection pressure. So this is what you get, a beautiful formula by Ewens that I'm trying to justify, not really derive. But look at that. So we have this factor here, which is essentially a normalization factor. And then here we have 1 to the B1, 2 to the B2. Remember, BK is the number of groups with exactly K individuals. And then you have this theta to the total number of groups. And then there is this factorial that we'll discuss in a moment. They are related to the fact that we don't distinguish between the groups. We just say there are B1 singletons, B2 pairs, B3 triads, and that's all. How do we get it? And this is known as, so you make a model for that. It's called the Chinese restaurant process. So this is nice Chinese restaurant somewhere in, I think in LA. What is a Chinese restaurant? Yes. No, this is not this is a probability. You have a sample of a population which is undergoing mutation reproduction process, no selection, of size n. Okay. Now, this sample, you bin this sample into putting together all individuals which have exactly the same allele, and you ask, what is the probability that I have B1 singletons, B2 pairs, B3 triads, and so on and so forth? That's it. There is no problem of fixation because anyway you have mutation going on, so if it goes to fixation, it starts again. Okay? Now, Chinese restaurant process. Why is Chinese restaurant process? Well, you have a large, I think that it works, sometimes it works like that in China. I surely used to work like that in Italy a long time ago. You have a very infinite restaurant, which is, say, close to close approximation picking, and has an infinite number of tables. Now, clients get into the restaurants, and then they have a probability theta to sit at a new table. Or they pick up at random one of the customers who are already in, and they sit, and he sits or she sits at the same table, right? This is actually not only for genetics. This is a process that is used in, for instance, it's used to model innovation. In a sense, theta is the probability of introducing an innovation. And there is also the fact that because you pick up a customer, you don't pick up a table, you pick up a customer, you have a tendency to aggregate. So it makes, it makes also a very null model for herding phenomenon, which large table is likely to get larger with time. Okay. Now let's solve the process. So at each we start, and then at each time you have, if you have n customers, then you have a normalizing factor. You have either n possibilities or theta, no? So you have n plus theta, you have to divide by n plus theta to keep a normalization of the probability. Now, each new table has a probability theta of arising, okay? And in going from k to k plus 1, each table gets a factor of k because it is chosen with a probability which is proportional to k. So that's the probability that the label customers sit at L tables of size ki, I is equal 1 to, so it's given by, so this is called probability of labeled. So you just have to imagine that I know the names of the tables and the names of the customers. And this is given by this theta to the L, which is this guy here, theta for each table. Then I have this product here, which comes from this normalization here. And then I have this factorial, which is the contribution of each table. It starts from 1, then 2, 3, until it reaches ki. Now we have to bin. Now first of all, this is label customers. We wish to remove the labels. So there are all the numbers, all the ways in which you can choose which customer sits at which table. So we multiply that there. And this is the origin of that factorial that we have seen. This we have already got, this we have already got. And then, so this is just a rewriting this stuff. And then finally, labeling the tables has introduced an overcounting. We are not interested in knowing which table is there. So we have to divide by the permutation of the labels of the tables. So this gives us this table permutation factorial. All right? Is that a good? So this is a nice way of looking at this. Ewens sampling formula appears everywhere. You will probably see it and it's very important in interpreting the data from genomical data. If you say people sampling alleles in a population, they get what they call a frequency spectrum, and this distribution of B's, say this distribution of B's is exactly what is called the frequency spectrum. So these are some consequences that you can work out. So one is the distribution of the number of segregating alleles. So you have this probability distribution. So this is you have number k of segregating alleles. When you increase your sample by 1, will be given by probability of having So this is, yes, K. So either you have a new customer, so the idea is that you have this restaurant, a new customer arrives, what is the probability that the number of tables remains constant or increases by 1? So this is just, here it increases by 1, so before it was K minus 1, now it's K. And here it remains constant, and this is what you get. You can solve this equation and get the average number of segregating alleles that also satisfies iteration like that, and this is the solution. So if theta times the sum from j equals to 1 to n minus 1, or 1 over theta divided by j, so this is just the number of the probability that it chooses, say, that k tables are eventually occupied, and the variance is given by this quantity here. And you can ask, what is the distribution of the number of singletons, which is easy to evaluate, no? And you see that from the distribution of number of from the average of number of singletons, you can directly have an estimation of theta. All these things, you have several statistics which all depend on theta. You can see if your sample can match with the same value of theta. You have also an idea of the uncertainty. So these are the observables that you can get. So this is distribution of the number of singletons for n equals 200, theta equals this quantity together with the asymptotic distribution of n equals infinity over 1,000, and the data over 1,000 samples, and this is what you get. You will see that it goes expected, well, it's not too bad, over 1,000 samples. Again, number of segregating alleles, and so it's quite nice also to simulate that and have some idea what happens. This is a frequency spectrum, so this is the average value of the number bk, also this number of groups with k individuals as a function of k. Here we have n equals 1,000, at theta equals 3.5. And you see that it goes first, this is logarithmic, and this is logarithmic, and the slope is -1. So it is, this is the behavior like that. It goes like 1 over k, and then it's cut exponentially at the value of the order of n divided by theta. So this is what you get. So out of this, you have, again, a way of evaluating theta pretty well by looking at the deviation from the 1 over k behavior. Effective population size.
Is that...
well, look at this. See, the dispersion becomes enormous because this is really there. You have either you have one sample, or you don't have it, so you have a great deal of dispersion at this level. You see, and this is again for say for values of the order of 1,000, of course. It's a finite fraction of 1,000.
Okay, thanks.
Now. So this is effective population size. What I said, population size. This is very nice in the computer. What happens to it in the wild? Well, usually what you get is that if you do this thing and then you have some independent evaluation of the mutation rate, you find that your numbers do not add up. Your numbers do not add up unless you finagle with the population size, and the population size you get out is much, much smaller than the actual population size, which is called, in this context, the census population size. So we say for humans, the census population size is a few billions, and the effective population size when you play this game is of the order of a few 10,000. So there's orders of magnitude of difference. And of course, this is telling us something. I will just point a few effects that go into that. First effect is in sexual population, you have that it happens quite often that the male population is contributing only a few individuals. Usually, say, female population reproduce and so on. Is the females each female has offspring, but the males who have offspring can be in a very reduced number. For instance, in birds, you can have this LEC reproduction mechanism in which you have that female congregate in some places in which there are different males in exhibit themselves, and then essentially only one of them is chosen for reproduction, right? So you have what is called a bottleneck, a reduction of the male population, and this reduces reduces strongly the effective population size. Another case, which is probably more important because it happens everywhere, is the fluctuating population size. If you look carefully in the equations, what enters is not the population size per se, but its inverse. So what is really relevant if you have a fluctuation population size is not the average population size, but what is called the harmonic average, which is the inverse of the average of the inverse. And this quantity typically is of the order of the minimum value. If a population goes through a bottleneck and then grows again, you have to wait for a number of generations of the order of the census population size for the equilibrium regime that I have described to apply. And because the census population is so much larger than the bottleneck population by definition, say definition of bottleneck, you have to wait a very long time. So you can see the structure long later. So this has some implications. If you like, say people have been using this kind of analysis, it's basically this kind of analysis to estimate what happened, for instance, to the human population in its evolutionary history. What they usually do is to make a model of the fluctuations of the population size and try to see whether the data are compatible with this model. There is no way to evaluate it directly. What you can do is something like a maximum likelihood estimation of what would. So you assume that there have been two bottlenecks and then intermediate expansion and then another bottlenecks, and then you ask, well, okay, I have a parameters, what is the age of the bottleneck, what is the size of the bottleneck, what is the size of the expansion, I evaluate these things by maximum likelihood, and then I get some history of the humankind that I can publish on Nature, right? And the other effect, which is very important, is that if fitness is non-uniform, then you have that the effective population size is reduced with respect to the census population size by the 1 plus the variance on the number of offspring that we have seen before. We have seen that for the case of M squared, this is just a pure rescaling, but if you have selection effects, then we have this very long tail. This variance can be very, very large. And I stop. No, I didn't show this graph. I will show you something, well, later on. Right? Okay, there are more questions? Yeah.
I could not ask that time when he was discussing about the e to the power sz.
Ekman?
Fitness equal to e to the power exponential of sz.
Okay, yeah.
Then you discussed about the distribution thing, no? The extreme distribution like Laplace or Gumbel distribution.
Yeah.
I just want to ask from that point of view that in some cases, for example, in some dynamical cases also, like logic dynamics, we sometimes take this type of extreme distributions like Gumbel distribution in logic dynamics in the perturbation case. So there the ultimate result comes in terms of the distribution parameter. Am I right?
What do you mean in terms of this parameter?
Suppose the standard deviation of Gumbel distribution is a parameter. So the final result in logic dynamics is dependent on the standard deviation of Gumbel distribution.
Yeah, sure.
So it is very important to know what distribution it is, because the result is dependent on the parameter. Am I right?
Yes, except that you don't know it. And probably it will be different in different conditions. It's very hard to know. So, and actually things are not so bad as they seem. And the reason is that you remember that there is the fixation probability becomes very small if the advantage is small, and then of course goes to 1 if the advantage is very large. So that means that very small advantage you can by and large neglect and treat as if they were neutral. And of course very large advantage anyway are not going to be very probable to arise. It's hard. So in a usual situation, what you have is a sort of intermediate value of the advantage, which is really relevant for the process you're considering it. And this turns out to be, well, not valid in all cases, but surely valid in a certain number of cases. I will tell you one because it's interesting. You can take a well-established life form, and the experiment was done by Andrew Murray in Harvard on yeast, and plug it in a different environment. Then by and large, in the beginning, what you have is that the adaptation grows more or less linearly with time by a regular accumulation of mutations, more or less with the same size of advantage. And there is a theory to that which is, well, more or less, goes more or less. Exactly for this effect, you cannot have a very small advantage because it doesn't go to fixation. You don't have a very large advantage because you simply don't win the lottery every day.
May I ask one more question?
So there's one. Let's first hear somebody else.
What is the average population here, means with respect to what, like males, females, or subpopulation size?
Here I only use the asexual population model.
This is sexual populations.
Okay, yeah, I just mentioned when you interpret this data in the wild, so this is an F. But remember, there was my disclaimer. I'm looking at, if I look at a small chunk of the genome, then I can treat the whole process as being asexual, but this effect is there. That means that, for instance, most of the male chromosomes do not enter the population if there is a lack. So this population the effective population is smaller than the census population. There are two things to that. So my model is asexual. I can use a sexual model also for sexual life forms. Then, for sexual life forms, one of the mechanisms by which the effective population is smaller than the census population is this phenomenon, which is, I use the example of lack, but it's pretty frequent in some way. Males have a chance if they can reproduce by and large in the wild.
Thank you.
Yes, please.
Yeah, that probability of... Okay.
No, yeah, you go ahead and then...
probability of fixation that you found out for a particular fitness advantage S, given the mutation rate mu. Okay? You found
probability of fixation does not depend on mu, depend only on S, which is the advantage with respect to the wild type.
It does not depend upon mu.
No, itself a probability of fixation. Sorry, because probably I use the same. There are two things. One is a conditional probability. If a mutation arises of advantage S, what is the probability that it fixes?
Exactly.
This is pi of S.
Yeah.
And then the P fix that I used is a probability that a mutation of size of advantage size S goes to fixation. This is P fixed that you're talking, and this is proportion, say, in the periodic selections case is proportional to mu.
Yeah, I'm just talking about that case where the probability depends both on mu and S.
Okay.
Okay, now I can find when this is maximum for a particular mu for what S mathematically.
Well, it's in the periodic selection case, it is proportional to mu. So there is no maximum with respect to mu, but there is a maximum with respect to S.
Now, is there any case where this thing depends both on mu and S, the function?
Yes, this is in clonal interference.
Yeah, that I am asking. There, for a particular mu, I can find for what S I am getting the maximum probability.
You sure?
Then in the mu S two-dimensional plot, I can find a curve which will give me the maximum probability of fixation. Am I right?
Yes, all right.
Yeah. Now, as we are studying the case of dynamics, you already told that there may be case when from bottleneck one particular category get fixed. That may be the case. So any dynamist may be not interested in the transient case, whatever is happening for 10 or 100 generations. I may ask, I am interested what happens after 1,000 generations.
Right.
So my tendency as a phenotype will be to have maximum probability of fixation. Am I right?
Yes, except that the distribution is not so sharp.
Okay, yeah. And now that S, selection advantage and mu, both do not change in time. Am I right?
Okay, let's assume that.
So in that S and mu plot, I have got already a curve that give me maximum probability of fixation. And you have given me a particular mu and S to do the model. From that picture, can I comment on what is going to happen after 100 or 1,000 generations? That's a pictorial view. I was thinking just.
It's interesting. We should look at it in more detail. Yeah, it's an interesting question. Yes.
Yeah.
See.
So what if variance is not finite? Then ME won't be defined.
Is?
What if the variance is infinite? What if it is diverging?
No.
So in that scenario,
no, average M is finite is equal to 1. So M square, average M square.
no, my question is, so what about the scenarios where variance is diverging? It's infinite. I mean, it's what if like it is of the order of population size, and generally you consider.
Yeah, it will be
very
of
large.
order of population size.
Yes. So
yeah,
in
but say from it works as if it were infinite.
okay, so in those scenarios, any won't be defined. I mean the.
Yeah, you have the Moltaski-Snitman coalescent. It's a well-defined process.
Okay, so one won't get Kingman's Coalescent in those scenarios. One will get MMC, multiple mergers Coalescent.
Yeah, you have a different universality class. You can treat it.
Okay.